In this video, we'll look at the final levels of protein structure, the tertiary and quaternary structure. Why are we doing tertiary and quaternary structure together? As we'll see, they're actually quite similar. They both describe the arrangement of a protein at a higher level than simple secondary structure. In both cases, the specific three-dimensional shape of the proteins results from assembling parts through largely non-covalent interactions, the van der Waals, salt bridges, and hydrogen bonds. And that's why tertiary and quaternary structure are often described together. Now, more specifically, the tertiary structure is the arrangement of helices, sheets, and loops within a single polypeptide. They're three-dimensional arrangements. Whereas the quaternary structure is the arrangement of several polypeptides into a protein complex that performs a specific cellular function. The tertiary structure is built from motifs to domains to proteins. What do I mean by that? When we describe how the secondary structure elements are arranged spatially, it's useful to define hierarchies of structure. And we'll explore this using alcohol dehydrogenase as an example. Alcohol dehydrogenase is an enzyme that we'll discuss again in a future video. Here is the full structure of active alcohol dehydrogenase, shown as a cartoon, with the helices in green, the sheets in blue, loops in gray. The smallest grouping of secondary structure is a motif, made up of several secondary structure elements that assemble in a consistent way. Like their names suggest, motifs recur in many different proteins with varying sequences. A very common motif, which we isolated here, is the beta-alpha-beta motif, where two adjacent beta strands are connected by an alpha helix. Notice how this allows the formation of a parallel beta sheet. One or more motifs can assemble to form a compact globular structure, a domain. Here, the C-terminal beta-alpha-beta motif, highlighted in yellow, interacts with additional beta strands and several other helices to form a domain. Domains are independent folding units within proteins. That is, an isolated domain can usually fold on its own and maintain its tertiary structure independent of the rest of the protein. Proteins often have multiple domains, which may then interact with each other to form the functional unit. In alcohol dehydrogenase, we can see how the domain we were examining, now in yellow, interacts here with a second domain, which features a Rossmann fold, as we'll see in more detail in a minute. The active sites of enzymes are often found in loop regions, and these loop regions can be shaped both chemically and structurally to provide specificity. Often, the active sites are in crevices at the interface between two domains, and these crevices are rich in loops. The active site of alcohol dehydrogenase lies between these two domains. Here you can see in spheres representation both substrates, NADP and ethanol, bound at the active site in the interface. Active alcohol dehydrogenase actually contains two identical protein molecules, as you can now see here. Its quaternary structure is therefore a homodimer of two subunits, with each one containing one active site at the interface of their two domains. Note how part of the dimer interface is formed by two interacting antiparallel beta strands to generate one large mixed beta sheet from the two parallel beta sheets in each Rossmann fold domain. So the arrangement of the secondary structure elements in a domain correspond to a fold. Like motifs, folds are found in many different proteins. For example, the Rossmann fold that we just saw is a very common fold that's found in thousands of proteins, and often proteins that bind nucleotides as cofactors.
What's the difference between a domain and a fold? Aren't they the same thing?
Not quite. Every domain has a fold, but some folds, like the Rossmann fold, appear in thousands of different domains in different proteins with different sequences. So alcohol dehydrogenase and glutathione reductase, for example, each have a domain with a Rossmann fold. Folds are used to classify protein structure. So the first order of classification in protein structures are three different big types of folds: the alpha folds, the beta folds, and the alpha-beta folds. Within alcohol dehydrogenase, we already saw an example of a mixed alpha-beta fold, the Rossmann fold. The Rossmann fold is a particular arrangement of beta strands and alpha helices, including several beta-alpha-beta motifs. This arrangement yields a 6-stranded parallel beta sheet that is sandwiched between helices on either side. An example of a mostly beta fold is carbonic anhydrase, shown here. Carbonic anhydrase catalyzes the interconversion of carbonic acid, the result of dissolved carbon dioxide, and bicarbonate. It is therefore a critical enzyme for maintaining pH balance in the blood. A common alpha fold is the globin fold. An example is myoglobin, which is shown here, the oxygen-binding protein in muscles. It releases oxygen under anaerobic conditions, which is important during strenuous activity. The globin fold contains 8 helices and is often associated with binding of a heme cofactor, shown here in orange. Hemoglobin is another example of the globin fold. Note how similar this subunit of hemoglobin is to myoglobin, despite sharing less than 30% sequence identity. These two domains share one fold. But unlike myoglobin, hemoglobin is made of 4 separate polypeptide chains and is therefore said to be a tetramer. This assembly of multiple polypeptides into one functional unit is called the quaternary structure of a protein. We often call these individual polypeptides subunits. These subunits can have different primary structures. Here, hemoglobin has 2 subunits of alpha hemoglobin and 2 subunits of beta hemoglobin. As we'll explore later in the course, the quaternary structure of hemoglobin leads to very important functional properties that are not possible for monomeric proteins like myoglobin. Quaternary structure can also be formed of multiple copies of the same polypeptide subunit. Alcohol dehydrogenase, which we examined earlier in the video, has 2 identical subunits, which are highlighted here in red and yellow. As one might expect, proteins with similar sequences have similar structures, but more remarkably, proteins with highly divergent sequences often still have the same structure as we saw for hemoglobin and myoglobin. It seems that there are a finite number of protein folds. New proteins tend to reuse existing folds as opposed to using a completely new one. This is why a classification of protein folds is useful. Shown here are just a few examples of roughly 1,500 folds that have been identified. This graph here shows the progress of structural biology over the last 40 years. The number of folds that have been determined in using crystallography or NMR or other structure determination techniques has risen, but has actually plateaued in the last few years, where almost no new folds have been discovered. And this is in contrast to the all-time high number of protein structures that have been solved. So there's a lot we still don't know about protein structures, even if we know the general fold of a protein. A lot of its function is determined by the details in its loops and specific side chains into how the protein functions. So the arrangement of proteins into multiple independently folding domains can increase their diversity. And that's because domains are modular. Gene recombination events can assemble new genes that code for new combinations of domains and therefore increase the diversity of the genome.
But what holds these folds together? Maybe that is conserved across proteins that have the same fold but with different sequences.